Hello, in this video of the bite-sized flu camp series, I will talk to you about vaccination, addressing vaccines for animals and humans. Over the last 2 years, we all heard a lot about vaccines. It was hard to avoid the subject with COVID dominating nearly every aspect of our lives. Some people are very skeptical about vaccines and refuse to get immunized, which often includes their children. For me, as an immunologist, this is hard to understand. There are many good vaccines against diseases which otherwise might be lethal or severely impact health and quality of life. The World Health Organization estimates that on a global scale, 2 to 3 million deaths are prevented by vaccines every year, mainly among children. So far, the biggest success in this field was the smallpox vaccine, which protected from smallpox disease. Prior to vaccination, smallpox killed about 1/3 of the people who contracted it. Thanks to the widespread use of the smallpox vaccine, case numbers decreased continuously, and in 1980 the virus was declared eradicated. Other successful vaccines in humans protect against polio or measles, which can also be quite harmful. For example, polio infections can lead in some infected people, about 1 in 200, to paralysis, which is sometimes life-threatening because muscles of the airway system are affected. There are also numerous vaccines for pets and farm animals. For example, dogs are frequently vaccinated against parvovirus, hepatitis, or distemper. Distemper, caused by canine distemper virus, has a 50% mortality in infected dogs. Cats frequently get vaccinated against feline parvovirus, leading to gastroenteritis, and feline herpesvirus and feline calicivirus, both causing infections of the upper respiratory tract. Similarly, there are numerous vaccines for farmed cattle, pigs, and chickens. For example, sows get vaccinated against parvovirus to protect them from abortion. In some farms, pigs also get immunized against different influenza A virus strains, protecting the pigs and the people handling them. This, as you may have heard in other videos of the bite-sized flu camp series, reduces the risk of the development of new viral strains, which may be more dangerous to both humans and animals. This is also an example of the One Health approach, where researchers try to develop strategies that protect humans and animals from diseases. However, pig vaccines against influenza are also an example of a vaccine which is not quite as good as we would like it to be. This brings us to the topic of different types of vaccines and what that means for protection arising from the underlying immune response. Influenza vaccines for pigs, and the same applies to human influenza vaccines, consist of inactivated virus particles. What does it mean? Viruses are grown in special manufacturing sites, and in the end treated in a way that their structure is maintained, but they are not able to infect a living cell anymore. Such inactivated virus particles are then used as a vaccine. Further examples of inactivated vaccines given to humans, next to influenza vaccines, would be the polio vaccine or the vaccines against rabies or tick-borne encephalitis. The advantage is that they are relatively easy to produce, also resulting in a low price, but the protection that they provide is often limited. This applies to both the duration of protective immunity and the breadth of protection. For several viruses, like the quickly mutating influenza viruses, good protection is only achieved for those viral strains which are part of the vaccines. Another type of vaccines are so-called live attenuated vaccines. In the past, their development was a try-and-error process, where viruses were grown in the laboratory on different cell lines. That means cells grown in culture dishes outside of living organisms. Often, this results in changes of the genetic code of the virus, which sometimes leaves the virus in a state that it can still infect humans or animals, but doesn't make them sick anymore. In this way, the immune system is trained to recognize those viruses which still circulate and can make us or the animals sick. Such vaccines often achieve a very good and long-lasting protection. There are several successful attenuated vaccines for humans, for example against measles, mumps, chickenpox, or yellow fever. However, for viruses that mutate frequently, like influenza or coronaviruses, this approach did turn out to be not useful or even impossible. Hence, during the last 30 years, researchers developed other strategies for the generation of new vaccines. One of them are viral vector vaccines, where a virus is manipulated in a way that it still infects the cells of a human or an animal, but does not replicate anymore, and hence does not cause disease. Such viruses get manipulated so that their genetic code also contains information from another virus, which is then recognized by the immune system and allows the development of protective immunity. The JADOX vaccine against COVID-19, developed by Oxford University, would be an example of such a vaccine. Other vaccines consist only of nucleic acids, which are put into protective lipid shells to administer them. This genetic code is read by the cells of our bodies, which start producing then single viral proteins. These proteins are recognized as foreign by our immune system, leading to its activation and subsequent protection when the entire virus enters the body. This technology is used by the COVID vaccines from Pfizer-BioNTech and Moderna. Vaccine development is an area of intensive research, and the COVID pandemic has shown us how important such kind of research is. Improvement of vaccines for animals is also an area of active research, with the goal to protect animals and humans alike, as envisaged in the One Health approach.